Good afternoon, friends. At the outset, I am thankful to organizing committee, Dr. Bansi Sabu, and Eli Lili, that is being headed by Mr. Rakesh Upadhyay and his entire team. So, I'm going to discuss something which is not frequently being discussed. GIP, what did we know and what have we learned? So, this is a relatively new topic and this is a sponsored talk. So, I'm a sponsored speaker for this session. So GIP is a forgotten molecule. All of us know about GLP, which came way back in 1980, so almost 45 years from now, 44, 45. And but this came a decade earlier. GIP, the elder brother, which is much discussed and much talked about hormone, and somehow it remained into excitement for a good number of years. So that is the story. And what these molecules do. The glucagon-like peptide, it is stimulated by glucose, and GIP is stimulated by glucose as well as lipids. So basically, this is the nutrient load in the gut that releases these two hormones. And there are many other hormones also that lead to the stimulation, but these are the dominant hormones, and both are polypeptide, and GIP is related by small intestine of K cell and the GLP from the L cells. The incretin effect is a well known, I think, way back when the first GLP analog came around 2000, we are aware. And what happens when IV glucose is given and when oral glucose is given, there is a difference in insulin level. And why it happens, and that is answered by the incretin effect, and these are the two dominantly known incretins, GIP and GLP. And then we learned over the decades after 2000 the another thing, that this is the incretin effect, and is there any difference in diabetics? So what was observed, that the rise of insulin after oral glucose was lesser, the insulin rise was lesser in diabetics. So we realize there is some defect at the level of incretin in diabetics. So what is incretin effect? And it gets impaired in people with type 2 diabetes. There is synergistic action of both the hormones, GIP and GLP, at beta cell of pancreas. And interestingly, in beta cell of pancreas, the receptors of both the hormones are there. GIP and GLP signal to a pancreatic islet to enhance glucose-dependent insulin secretion. So everything is glucose-dependent. That is another interesting part. And they are receptors. This is the hormone, and GIP, GLP gets attached into this. GIP and GLP together have synergistic actions on glucose-dependent insulin secretion. And if you look carefully to this, this is the normal cell line. This is GIP and this is GLP. So if you look at the after, this is on healthy individuals, healthy volunteers. So the insulin level after GIP and GLP were there, but if you give two together, they are much more than the combination of two. So there is an enhancing effect of both. And why this happens, I think next couple of slides, I'm going to talk to all this. How it acts. When we eat food, there is a nutrient load, like we have just had our lunch. So there is a nutrient load, and there are two levels of connection. There are glucose receptors at the level of beta cells. So it stimulates the beta cell to secrete insulin, and at the same time, it also, there are receptors of GIP and GLP at the beta cell. And from intestine, from I and K cell, there is a release of GIP and GLP, which gets stimulated to the receptors presenting on beta cell, and from there is action and eventually the release of insulin. So insulin is released by two modes. One mode is the glucose load and other mode is through the GIP and GLP. So there are two modes of action, and let us see what is the contribution of each one of them. GIP GLP increase in response to oral glucose. So if we look, this is the GIP response, GLP response, and this is the GIP response. So magnitude wise, a little more. GIP accounts for 2/3 of incretin effect in the human. So if whenever we eat our meal, there is a glucose, and it directly stimulates at the level of beta cell of pancreas and leads to release of insulin. So this contributes around 33%. When comes the long known GLP-1 receptor agonist, and its contribution is around 22%. And then simultaneously from K cells of small intestine, there is a release of GIP. It contributes to 44%. So if we look magnitude wise, there is a almost double difference, 22 versus 44. So the stimulatory effect of GIP is probably much more. And there is an enhancing effect of both of them also. So this is the way it acts, and what is important to realize that the GLP, which is much more known and a very powerful molecule, which all we have been experiencing over the decades. Now we have oral molecules also. So, but the GIP, the magnitude is much more. The potential to release insulin, the potential to control diabetes. The GIP action is influenced by the receptor concentration, and what is interesting is that the beta cell of pancreas, we have receptors for GLP, and on the alpha cells also we have the receptors of GLP and GIP both, and adipose tissue we have only receptors of GIP. So here lies the difference, because if you look at carefully the alpha and beta cell, we have GIP and GLP receptors, but on fat dominantly the GIP receptors. So that makes the story little different. Receptors are present in other parts of body as well, other parts of brain and body as well. So GIP and GLP, what happens to glucagon release in response to oral glucose? GLP, as we know, it stimulates increase the insulin and reduces the glucagon. GIP, it increases the glucagon. So why this happens in a normal individual? There has to be a balance between two. The balance that is between insulin and the glucagon and blood sugar. So similar balances between GIP and GIP. So in healthy individuals, GIP stimulatory effect and GLP inhibitory action regulates the baseline glucagon secretion. So if this is the healthy individual, and this is the isoglycemic clamp, and the and look at the glucagon secretion. So this is keeping a balance depending on the insulin level, depending on ambient blood sugar level. What are the actions of GIP at adipose tissue? And this is the key thing which differentiates the action. The preclinical studies have shown GIP may improve insulin sensitivity. Activation of GIP receptors in adipose tissue enhances insulin sensitivity in adipose tissue. There is a improvement in adipose tissue functions. And what exactly it does, it enhances the hormone, the lipase activity at the level of fat cells. And what is the effect? Eventually all the fat in the circulation gets stored. Fat which is dominantly in the form of triglycerides, gets stored in the fat cells. So the circulating fat levels are more reduced, and the triglyceride is one chemical which is responsible in bringing about insulin sensitivity. So more is the triglyceride, more is the insulin sensitivity. Less is the triglyceride, less is the insulin sensitivity. And not only this, it has been observed that in healthy individuals, GIP increases adipose tissue blood flow, which makes the fat cells healthier tissue. The more fat is mobilized from the circulation, the more circulating fat and gets stored into the adipose tissue. Another thing is noted that you can ask if the fat cell is mobilized, fat cells would be thicker. No, but at the same time, it has an inhibitory effect at the level of brain. So the intake at the same time is also reduced. So the fat intake is also reduced. So there are a dual contribution. One hand, the fat is mobilizing into the fat cell. Other hand, by virtue of decreased intake of food, the calorie intake, the fat into the circulation also goes down. And there, if you look at the slide, there are hyperinsulinemia and hyperglycemia along with GIP. So this is the slide. So hyperinsulinemia and hyperglycemia both are the stimulatory effect. On top of it, if GIP is given, the blood flow is increased. And what it does, so GIP plus hyperinsulinemia plus hyperglycemia increases blood flow, glucose uptake, and free fatty acid re-esterification at the adipose tissue. And what it does, it increases fat utilization, decreased release of free fatty acids. And by virtue of this, there is a reduction in decreased blood glucose and there is an increased free fatty acid re-esterification. More fat goes into the adipose tissue. To sum up, enhanced insulin secretion in response to oral glucose as compared to intravenous glucose is known as incretin effect. So incretin effect is a very old effect which we know, and the incretin effect is reduced in people with type 2 diabetes, and 2/3 of incretin effect is contributed by the new molecule GIP. GIP action is mediated by receptor concentration, one at beta cell of pancreas and other at the subcutaneous white adipose tissue. So its function on adipose tissue is very important because it mobilizes the fat, it reduces the ectopic fat, and by virtue of this, there is a reasonable amount of alteration in lipid metabolism. Beta cell of pancreas, GIP causes potentiation of insulin release. Adipose tissue level, GIP causes better lipid uptake and increases the storage capacity of at the level of subcutaneous white adipose tissue. This, however, does not drive obesity if energy balance is maintained. This is what I wanted to say. And to sum up, it works at the level of brain. The GIP action is dominantly it reduces food intake, while GLP it reduces food intake and increases satiety. At the subcutaneous white tissue, increase insulin sensitivity because of mobilization of free fatty acids, triglycerides, increase lipid buffering capacity, and increase storage capacity. More fat from the system moves into the adipocytes. At the level of pancreas, GIP activity increases insulin. Both the hormones increase insulin. GLP reduces glucagon and GIP increases glucagon, but net is the more or less same. And at the level of stomach, the role is dominantly by GLP activity, so it reduces gastric emptying. So this is the small briefing of a new lesser discussed hormone, the elder brother, which is I think will be launched in the future, and we are in a process of learning this molecule. With this, thank you so much for your kind attention.